All right, so part two, translation. We are now an mRNA strand that has entered the cytoplasm. The mRNA strand has left the nucleus and it has entered the cytoplasm. Translation occurs in the cytoplasm on a ribosome. And the tRNA, that is this carrier molecule here, tRNA, the T this time stands for transfer. Remember that the M in mRNA was messenger. Transfer RNA is these little triplets here, and they are going to bind to these triplets of mRNA. This triplet we can call a codon, and this triplet here we can call an anticodon. So for tRNAs, they are anticodons, and for mRNA, they are codons. Just the terminology for triplets in their respective RNAs. So another thing to note is that this gap, all these gaps here are just here for illustrative purposes. It's just so that we can make it really clear that the binding is going on between triplets, that the codon and anticodon are what are complementary to each other. And you'll see that the rules that we established in the first video are still true, that U is still binding to A is still binding to U to A, and over here, C is still complementary to G, C to G, C to G, like that, okay? So that same alphabet that we established before, where A's bind to U's, G's to C's, if we go all the way back to DNA, we'd be thinking about A's and T's, wouldn't we? But remember that in mRNA, we have uracil, and in tRNA, we have uracil instead of thymine. So these gaps are not really there. This U is next to the C, but it's just a nicer way to look at it. Okay, and this is a ribosome. Ribosome is just one of the organelles in a cell. This is again simplified. If you really want to know what it looks like, it's more like a delicious hamburger. And the chain goes right through it. Okay, and the chain of mRNA goes right through it. But that's not the most important thing just now. The most important thing now is saying, what has this all got to do with creating a protein? Well, hopefully we'll have noticed by this point that the carrier molecules of tRNA, the transfer RNA, are all bound to these separate amino acids. Okay, so amino acids, what are they? Amino acids, there are 20 of them that we use to make up human proteins. There are 20 critical for human protein synthesis. Okay, and they are found bound to tRNA in the cytoplasm. Bound to tRNA in cytoplasm. This is our tRNA here, our carrier molecule. It's carrying the amino acids. And a chain of amino acids is a polypeptide. That word poly again, polymerase, polymerization, polyethylene bag. It just means many. A chain is a polypeptide. Okay, lots of peptides. You can call an amino acid a peptide. For example, you've got these carrier molecules here. So, for example, the tRNA that is adenine, uracil, adenine again, that carries tyrosine. And it will always carry tyrosine. And that's good, because that means that if AUA always carries tyrosine, that means that there is continuity, that the DNA and the complementary mRNA, every time we create this little triplet, this little codon, that we can be sure that we're going to create a that in its place is going to be an amino acid of tyrosine. So that's the code for tyrosine, essentially. This is the mRNA code for tyrosine, and this is the tRNA code for tyrosine, which is complementary to it. So let's have a little look. So what actually happens in translation? So in translation, tRNA binds to mRNA. Binds to, and it brings along its, it carries along its amino acid. Let's just make sure we've got a note of the codon-anticodon relationship. A triplet of mRNA bases, a codon, binds to a complementary triplet of tRNA bases, and that is our anticodon. Okay, and as this complex moves through the ribosome, moves along the ribosome, moves through the ribosome, the amino acids are linked together as a polypeptide chain. So, as this complex passes along the ribosome, the amino acids are linked as a polypeptide chain. As this complex passes along the ribosome, the amino acids are linked as a polypeptide chain. You can see the first of this chain being made over here between leucine and arginine. And this here is a peptide bond. Okay. One thing that just illustrates my point about the way that the template is conserved is, look, we've got two molecules of arginine here, two amino acids, arginine, and you'll see that they've got the same code. GCC makes arginine. GCC makes arginine. Okay, so that it's not done in any sort of shoddy or haphazard way, and that enables the template to make sense and for genes to code for things reliably. Because let's say we're growing a new, maybe 1,000 skin cells to replace a cut on your hand. You don't want some of them being brown, some of them having hair, some of them, I don't know, growing nerves, and some of them growing muscles, you know? You want them to be exactly the same. So that's why the template is conserved and the codons always code for the same thing. What else have we got? So, yep, just to say that this sequence of amino acids, getting lazy just writing is the primary structure of the protein. Later on, it will get folded and it will combine with other molecules to become something quite complex. Let's say that in the protein synthesis, there's a lot of cells coming together, and let's say they're making a red blood cell. Well, a red blood cell needs 4 heme groups, doesn't it, to hold on to the oxygen. Heme is quite a complex protein, and there's various foldings and different quadrants coming together and all this difficult stuff that's a bit above our level. But at the very start of all that, you need a primary structure, and that's what the sequence of amino acids is. Since this one's been a bit quick, I suppose I'll do a little recap on the whole thing. Oops, that's not it. Where's my other sheet? Yeah, okay, so we've looked at the attachment of the mRNA to the ribosome. Okay, that's the first thing that happens after we leave the cytoplasm. The mRNA attaches to the ribosome. We're at pains to discuss the different names of the triplets. You've got triplets of bases in the mRNA are called codons, and the triplets of bases in the tRNA are called anticodons. The transfer of the amino acids to the ribosome by the tRNA, that's why the tRNA is called the carrier, okay? Because the amino acids aren't going to make their way there in the correct order by themselves. That is just a massive amount of chance and random dumb luck. It's never going to happen. So the way that we get these amino acids that are floating around the cytoplasm, the way we get them to line up how we want them to line up so they can form the primary structure of a protein, is that they're attached to tRNA, which binds to the mRNA. The mRNA is in the right order, therefore the tRNA is in the right order, therefore the amino acids are in the right order. And finally, the ribosome's big job is to link the amino acids to form polypeptides, and you get what's called a peptide bond. So once this ribosome has moved along here, and it gets all the way to over here, you're going to have a lovely chained up polypeptide, which makes the primary protein structure. I hope that was helpful. If you do have more questions, that's fine. Send them in an email. Hopefully get back to you quickly, but if it's the weekend, we'll see. I'm sure you'll do great in your test, okay? Best of luck. Goodbye.